So today, what we're going to be looking at is the cross-link analysis with Kojak. You'll also go through the resources that we have for SWATHAtlas and SRM-Atlas. And then we're going to finish it off in the afternoon to take you through a data analysis process using SWATH data. And this will be done by Sam, but you won't have the software there. So the rest of the components, you will have the software and you'll be interacting with some of the programs that we've got. But apart from that, this will be just a viewing exercise so you can see how SWATH data can be processed and you can get to see some of the results. So this is where we are. So we're going to be doing most of the interaction stuff this morning. Then after lunch, that's when we'll do some of the SWATH analysis as we finish off the day. And we really have so now you've learned everything about the TPP. You've been listening to all of the instruction, and then now you know why that we're doing things the way that we're doing and having this statistical analysis. And once you get through all of this, you've come up with a list of proteins, and you really want to use those ones to now go back and do all these quantitative analysis, and that's what we're going to go through. And so with Kojak, that also utilizes this approach as well, and then outputs that data. So as you've learned with cross-linking, that you know that the whole goal here is to try and understand this three-dimensional process of which the proteins are actually coming together. And by having cross-links, you can then get this spatial arrangement. And if that peptide is linked to that peptide, they must be very close together, and so you can understand how that topographical location is really done. But to do that, you really need to understand how you can get these mixed peptide sequences analyzed. And as you know, that when you're looking at a single peptide, it's quite easy. It's not too hard to actually do, and you can get this done automatically. But when you have numbers of peptides together, 2, even 3, then it becomes very complex. Sam will go through all of the aspects of taking the data that's being collected, how to collect that data, and then process through the SciEx software. So he's going to go through, because this is the software that you'll get if you're running an AB SciEx instrument, and creating these libraries and analyzing with those libraries and utilizing some of the resources. And so we have the SWATHAtlas.org that you can download libraries to actually use. And we also encourage you that if you're making a library and it's a unique library, that you would submit this to SWATHAtlas as well and become part of that community. And so that's we'll be going through. And so in the end, what you'll get to see is how to process these complex profiles where you get multiple peptides being selected and then fragmented, and then how to actually take that information and get good quality data, and how do you actually understand that it's good quality data as well. And this is the type of profiles that one would see for a particular protein, that you see that they're very much like SRM peaks, but you've got all of these other transitions from different peptides coming through. And so Sam will take you through those results and work out, you know, what constitutes a good assay, a SWATH assay, and then how do you actually detect those assays with confidence.
So I'd like to give you today, right now, a short tutorial, whenever remote desktop connects, about the SWATH analysis. I'll do that myself because this analysis takes rather long time, and so if we do that, it would be like, okay, or we should have started like on Thursday evening doing something, and then we could continue like Friday and so on. So that's why I just like have something sort of like pre-cooked, and I would walk you through them. But like this, I can walk you through like all the different stages. And if remote desktop doesn't want to, it doesn't matter. I'm going to do that with the PeakView software. This is a commercial software of SciEx. I'm not entirely sure if that comes just with every triple TOF and so on, but I'll do this. So I'm gonna show you the analysis the targeted way, not the untargeted type of analysis. I'm going to use PeakView because it has a nice graphical user interface. As long as you do that with like just individual peptides or proteins that you know you're interested in, you know what you should look for and so on, that's fine. As soon as you want to have a broad picture, what you end up with is just essentially a table with all this information, and you would have to walk through that. This is also rather like, it's like if you have a shotgun analysis with a long list and you have to make sense out of that. So there it gets a bit complicated. But there I can't really help you either, because I mean, you know the question you were asking, you know your experimental setup and why you chose it that way. So this is this, and there the other tools. So like SWATH, Prophet, and OpenSWATH are just fine if you get a table. You don't need these fancy user interfaces. OneOmics is essentially peak view on the cloud, and has also like, I mean, it's from a company. They have to make you buy it, so it looks nice, and it's very user friendly. I haven't used it yet. I just saw like some people using it. Spectronaut is from the company Biognosis that also sells the IRT peptides, and you have to have these peptides spiked in order to use the software. However, the software is free, so I mean, your choice. So this is a short rundown. The way I'm going to present it. Usually what you would start with is either get a library from SWATHAtlas or make it yourself. I placed it down here because then we can load a session and to like then look at the results, and that takes some time. So like this, we use the time a bit more efficiently. But usually this is the first, that would be the first step. I'm going to show you now just from like assuming you have the library and assuming you have the SWATH files. I just have a trial license, so usually there's a peak here. I don't know why mine doesn't show up, but yeah. You go here to quantitation. There's SWATH processing. And then it lets you choose either a protein pilot group file. This is, so protein pilot is the search engine from SciEx. And this is kind of the result file that they give you. They can, like this, you can just like import it here, which is a neat tool. Otherwise, you can import text files, and it has to have the ending txt. We're gonna do this because SWATH Prophet doesn't make group files, they just make text files. And they also allow you to upload MC-identML files. I haven't used that yet, but yeah, that should be also a tool. I think that works better with, I think Mascot is able to generate that, and so if you have Mascot output, you can do it that way. So I selected this. We're going to work with an MTB data set, so we could just selecting a library. If you then, on the next window here, you can choose if you want to import shared peptides or if you just in general should exclude them. I usually leave everything in, but I extract everything anyway because, I mean, you have the information. Afterwards, you can, on one hand, the statistics get a bit better if you have more, and on the other hand, you can always delete afterwards. But if you exclude some things beforehand, you have to like redo the whole thing again, and because it takes some time, you rather do it the first time. Down here, you could translate your ion library. So the ones that are on SWATHAtlas are all light isotopes, and here you can say I want to convert them, and I have a labeled sample with like all sorts of labels. You should know that some of them, so like MTRAC and dimethyl, they modify the peptides. And so if you have a library from SWATHAtlas and you want to use it like that, it might be that it doesn't work that well. The reason for this is that these tags usually affect the retention time and may affect the fragmentation pattern. So I haven't tried it, but you can. The other thing, like converting it to SciLac, works rather nicely. I mean, it isn't that hard, and but it only generates you a heavy library. It doesn't generate the heavy light library. But we're not doing that. We don't have SciLac mycobacterium. Okay, now it loads, and this is just the assay library, so you might get a hand on that, how long it might take if you load other things. Done. Good. Now the second thing you need is the SWATH files. SWATH files. So I'm going to select. We're going to look at all of them later, but just for purpose, you can select them, and here you see. So you can select them and transport them over there. You can go to multiple like folders and select them from like, no matter how it's organized. You should know the more you load, the longer it will take. It also, the triple TOF, I don't know how many of you have access to one. It allows you to have like one WIF file, but like multiple runs in there. That's, you can load these as well in here. I mean, well, it's their software. You've seen once you've loaded everything, you specified everything, you get here a pane with the proteins in, that are stored in your library. Here's a pane with the peptides that is in there. This one here is shows you the traces that you've got. And this one here is the actually acquired MS2 spectrum at where that blue arrow is. And this is, if there is a peak group, like a nice looking one, the apex of the peak group. In this particular case, the library is the retention time of the library is normalized and is in IRT space. That one starts at -50. So they're all shifted. That's why you don't see here anything. So you have to do the retention time normalization in order to get that right. What else do I have to do? Oh. What you can see is if you go here on that drop-down menu, you see there are these 3 files that I've selected. And if you select 50, there will be 50 files. You can go through them. You can pick just another one, and then you see it'll take a while, and then it updates that one and this one in order to if you go to processing setting, you can see the processing or the options that you have. This is on one hand, you can limit the peptides and the transition that you want. You can have a peptide confidence threshold. This is useful if you use the library directly from Protein Pilot, because there you can like say, well, I'm a bit more stringent and less stringent. Since with SWATHAtlas we already did the filtering, and most of them are for like 1%, an error rate of 1%. You can just import everything. SWATHAtlas libraries don't offer to like use that as a filtering. They don't. It doesn't specify there how confident you are for that assay. You can specify a threshold for the FDR, and this is important. If you then export the results, it will only include assays that meet that criteria in at least one of the data files. So you could, obviously the higher you go, the more it will be exported, the larger are your files, and the harder it is like to walk through it. But yeah. You can, again, exclude the shared peptides. You can also exclude modified peptides, because you might just want to look at the unmodified ones. You can also fix a rank. I'm going to say something to the rank a bit later. And down here you have more mass spec related choices. One is the retention time window. We'll cover that later too. and a mass error for the fragment ions. So what's the mass error that you tolerate? Both of these are the entire window. So a retention time window of 8 means it is the center of where you would expect it to be, plus minus. So this one here means it's the retention time where you expect it to be, plus minus 4 minutes. And the 75 ppm means it's plus minus 37.5 ppm. This one here is default. I haven't changed that. And the retention time window I'm going to cover later. Let's see what my cheat sheet says. Initial parameters. So, as I showed you, one additional thing. Here it says number of peptides per protein. If you set that to, say, for instance, 10, if you happen to have 20 assays for that particular protein, it will filter, or it will only show you the peptides rank 1 to 10. The idea behind that is, again, if you have very large libraries, like the human library, it might be useful to just look at a few peptides. The problem is, how do you rank the peptides? Right now, it's simply the first peptide in the library from the top is number 1, and then number 2, and so on. But that doesn't have any meaning in terms of like how relevant they are for you, for your question that you have, or how easy it is to detect them, or how unique they are, or whatever. So it's just random. That's why I have here a rather high number, in order to like be kind of sure that all of the assays are included. The other settings you can. Same is true with the transitions. There goes the same thing. So now it will only use 6 ions and not more. There, however, you do have the relative intensity, and you can select them based on that. That's how PeakView does it. That's why you can load in huge libraries with like 40 ions per peptide if you want. So that works actually quite nicely. Yeah. So it might be quite good for you to know, like, how do you actually select, or what's a good number of transitions? And there it's, I have here a plot for a library that I used once, and just looked at the number of identifications, either on precursor or peptide level, these two, or the protein, which is on that level. How does that number of identifications relate to how many transitions you have? And I found for that particular library a maximum of 6, or 6 transitions to be working best. You can explain it that way, that the more transitions you use, the more specific you are. But also, the more transitions you use, the more likely you are that one of these transitions is actually no longer observed. And also, you get fewer assays in there. So I told you, I think, Saturday, that in that particular case here, I only used assays with 6 ions. If they had fewer ions, then I haven't looked at that, at these peptides. So yeah, using too few transitions makes it easier for a decoy to be a match. So 3 ions to randomly appear and randomly get a good peak group is more likely than 6. And yeah, if you use too many, it's harder for an actual target assay that all of the transitions are observed. In case of the retention time window, it's something similar. In this case, I use the same library, and here it's again the retention time window in minutes, and you can see the wider you make the retention time window, the fewer identifications you get, and this has mostly to do with that. Okay, the text is fine. This has to do with that the larger the window is, the more opportunity is there for a decoy assay to actually get a hit, to actually have a nice peak group there. If that window is too short, you're obviously going to exclude the target assays. And then if I go lower, say to 4 minutes or 2 minutes, you will see a significant drop. You can estimate what kind of window you should choose by just looking at a few peptides, like across your runs, and then see the deviation in the retention time, and then say, okay, that's maybe a good window. Or you can just say, choose a subset of your library and do the extractions at different window widths, and then determine it like this. Good. So now let's do. Okay. So if you've seen that here, the retention time doesn't match this one, and this and this one here is the IRT retention time. So that's why it's, say, about 50 minutes off. Also, since there are negative values, you see this one here. Somehow the graphical user interface doesn't like negative values in there and just doesn't display that. What happens in that case, if I click on it, so for all the other assays, PeakView would show you up here the retention time window you selected as specified in the settings. If it doesn't have a retention time in there, or if it's a negative value, it will extract it over the entire range of the chromatogram. That's why it also takes quite a while for it to display. Come on. But yeah, it still has the retention times in there, and you can, as long as you do the normalization, and as long as it shifts all the expected retention times after normalization in a positive space, so above 0, it's fine. And you can also save it and open it, and that should work. But just initially, don't be surprised if here is something empty and it suddenly extracts it over the entire range. You see it. So in this particular data set, I've spiked in synthetic peptides. And so we're going to use these to normalize the retention time. And this is actually quite simple. You just select, so in my case it's just one protein, but in case you want to add a peptide to your, if you want to select that peptide to normalize the retention time, just click on that little button here. Now I'm going to do that for these 10 peptides. You can also see in the trace field up here, you kind of see that there are no peptides there. This is not how good data looks like, so that can already give you an impression for like how a wrong result might look like, or just as an example, simply because it looks at the wrong place. Come on, just one more. In case you do the normal, in case you don't have spiked in peptides, you have say endogenous peptides that you want to normalize with. I recommend you to. Okay, let's go ahead because that takes a while too. So if once you're done adding the peptides, you can click here on RT with that little pen, and now you see here the assays. So this one here, this is the assay. See the charge state, essentially the precursor ion M over set, the fragment ion M over set, and this one here is the retention time. And if I scroll down here now, you see that guy actually has a negative value in there. So before you haven't seen them, but here it is allowed and it will use them. And so I would say let's just start that. that will take a while too. So in case you have endogenous peptides, you want to use these, I would recommend you to duplicate these assays and change the protein ID to something like RT protein, because then if you use that software, you can just specify, you just go to that protein and then pick all the peptides like I did here. The ones coming down here, these are just peptides with another charge state. These are the same ones. If you want to know, so if you do not have any peptides, if you have just a lysate without spike-in peptides, what you can do is you download the library or you generate the library yourself, you make a text file, and then you just delete all the values from the retention time columns. And then you extract. And then from the result files, you just look whatever looks good. You won't identify as much as you will with the retention time specified in there. You also will use the entire chromatogram, so it will take a much longer time to extract. But in the end, you will find essentially these assays that are identified very easily. So you don't need that value to identify them. And you can check if the retention time is okay, since you have the library and you know about at which time it should elute. So like this, you can kind of easily define your own peptides, make your own protein, or retention time protein. I would recommend you, whenever you do this, that when you have all of them, you just go through them and have a look and make sure that these peptides actually give you a good result. And I need that because I want to show you something like how you can correct or edit it, as well as control it. Are there questions? Yes.
Is this calibrating the retention time?
Yes. So what it does right now, that's a good thing. What it does right now is it takes each of these assays and extracts it over the entire chromatogram. It doesn't use an as sophisticated scoring system as it will afterwards. So that's why there are sometimes some issues. So that's why you need to look at it later. It's also faster if you only do 2 or 1, or you don't have a 3-hour gradient. Are there questions so far? One thing you can do as well with the retention time with spike-in peptides as well as the endogenous is actually look at which ions work best and like optimize them. Because in some cases you will see it doesn't really like interferences, or if the peptide has a really broad peak, it might vary with the retention time from like one file to the next, and that gives some variation to the retention time calibration. But you also see here that there's quite some space here as well as here. So with these synthetic peptides, although like well spaced, and originally they were generated for, I think, SRM, so like shorter gradients, not like here like 3-hour gradients. It might be worth to like pick some more. And if you remember the slide I showed from the endogenous peptides, they were like nice and evenly spaced, but they were also like 41. So yeah, we're almost there. It does that. Yeah, you see, it will be faster at the end because it does it like through the chromatogram, and so now it's probably either in the wash or at the re-equilibration step, so there's not that much signal. I don't think it is as interactive, but yes, and so from the it's nice once you get to the result, like result display. There you go. And now you see why I've chose these 3, because there's an issue. So yeah, Uli knows that. she had her time with it. So here you see, in this range, it now updated the actual retention time to like what the actual retention time is. Before it was from 0 to 1, and you can see here these are the 3 files. So the blue, the red, and the purple one. You can go down here and select a different one and then see how these different linear regressions are. This is all it does, by the way. It's just like, this is what it should have as a retention time. This is what it actually measures. It takes an easy linear regression, and that's it. What you see now is, go back to blue, you have an issue, or up here you have quite a spread. Down here it deviates as well. But what I also have to say is in these kind of regressions, the endpoints have quite a big effect on the entire thing. So we're going to have a look at them. And you see, do I only have one of them? Oh, damn. Oh, it doesn't matter. So I thought I added the triply charged of that peptide as well. That's actually the more intense ones, or the one that actually performs well. So in this case, you see here's some variation. What you can do interactively is sort the retention time column from like the first to the last. And then you can click that one and just with the delete button, delete them. And if I completely deleted it, that one is gone. You see, yeah, what do you see? Where is it still? No, it's not there. So now you saw it already corrected the regression, and now it looks much nicer. So that's all it was. So that will happen more frequently if you have endogenous peptides, since they're just not quite as good as IRTs or spike-ins, but they still work well enough. Once you've done that, you can just click apply, and now it will update the now it will update the retention time. And what you can see now is, so that one has 15 minutes, and all of them have a value in there. So we should be good to go. So now all the peptides in there should actually have an expected retention time. These fields here, these are then filled out once you do the extraction. In order to start an extraction, I usually check again if the settings are right. And then I would extract that. I haven't done it here, but trust me, right now it just uses the library as a name. That's a bad idea, because you're going to use that library for like some other extraction or do it slightly different, and in the end you don't remember which one is which. So it's worth, or at least what I've been doing is specify like which data you used and the settings. So like the retention time window, how many transitions, and sometimes the mass accuracy, but I kept that fairly, or usually constant. And we have to change the name. So the way it could look is that I'll give you an example. So I used we had a gross curve data set. We used 8 minutes, 6 transitions, and 75 ppm. Something like that. And then if you click Save, it'll take some time, and then it'll show you a window like with the progress. I'm gonna cancel that as soon as it shows up because, yeah, we will be here tomorrow. Okay. Instead, what I do, come on, if it lets me, I will load the result file, the result section from that. In that case, I've used all the 9 files that you saw before, not just one set, but all of them. While that is loading, we're gonna go here. So if you type in SWATHAtlas in Google, you'll reach it. Otherwise, it's swathatlas.org. It's not that hard. This is where you can download like entire libraries. So like similar to it has very similar functions, and it will also look kind of similar to the SRM atlas. However, here you don't download like peptides for individual proteins or like even individual peptides. You download them for like organisms, more on that scale. We also included some nice features that are kind of specific for SWATH. If you want to download, or where I got the library from, is just click on download library, and then you see here nice tables that specify you. So this is just a name for the library and an accession, and it tells you which instrument was used. You might have a preference for triple TOF if you have a triple TOF yourself, or if you've done the analysis on a Q-exactive, you might want to use a library on that one. It has several formats on there. It also shows the size and has links. The formats that we currently have are the normal Spectrust library, and in this case you have both the consensus and the raw. This is helpful for, say, for instance, the consensus you can use if you just want to add that to something. Say you're analyzing a virus and want to study now the virus in what happens to the host when the virus attacks or something, but you only have the library for the virus. You could download here the consensus Spectrust library, add the virus library, and then make your assays like that. The raw library here is useful if you're on using human yourself, and you think if you make the assays, it's better than if we do that. And like this, you could like combine the libraries a step earlier. You have down here different kinds of OpenSWATH libraries. These ones here are just specifications for the Windows settings that they've used. You can either download them as a text file, so a CSV in that case, or a TRAML file. TRAML files are standardized, and this is the one that you should be using. The problem is, if you look here, the CSV file has 221 megabytes, and the TRAML file, which is the exact same information, that's like over a gig. So you know, probably no one is doing that. There are also decoys added in the, or you can add or use these where they're already added. These are the ones like similar to the shotgun searches, where you have the peptides like pseudo-randomized. And this is OpenSWATH. Down here you have one for peak view. This is essentially you have all assays, all ions, and no decoys. That's pretty much the same thing. Peak view does the filtering for you. It filters the range around for the selection window. It filters that for you. In case of OpenSWATH, that specification here about the window is like these ions are excluded. So that's just a short description of the like how to look at that table. If you scroll down, because we don't want human and yeast, we want Mycobacterium. You also see here some information, like a title. There's a publication that goes with that as well as a data set. So you could, theoretically, you could go to the data and make the library yourself. But yeah. Down here some statistics. So like coverage, 99% of the proteome, or 3,984 in that case. So it's fairly high coverage. In case of the human, by the way, this is only the expressed proteome, essentially. So it's only 10,300 proteins, but it should allow you to do most of the projects you would want. Down here, you could either just download the PeakView library and do the same thing what I did. I mean, this is what we're going to look at later on. But I'd like to show you here that you can also customize these libraries and say, like, it's nice that you made these libraries, but they're too big, or I want to have them specified the way I want. You could also, on the first page of SWATHAtlas, you can, on down below, there was like a direct link that will get you here, and then you can just pick the library yourself if you want. You could name the output file test2. Test1 was this morning. Protein, you can filter for proteins, and since we are looking at the DOS-R regulon, so at some proteins that are regulated by transcription factor later on, I'm going to just specify these. In that file, this is that file, you have here the protein identifiers that are used in the library. If you're not sure, just download the library, have a look at it. Like, just look at the first few lines of it and see how they're specified. Usually, so in case of human, there are UniProt identifier. In case of the MTB, there are these RV. These are the systematic names of the genes. So they're named from like 1, which is somewhere on the genome, and then 2, 3, and so forth and so on. The C stands for complementary, so that's just the other strand. And then you can pretty much build your genome. And so that's why it's useful to have these. But yeah. So you have these identifiers, and then you can filter them. So like now, you see there are 34 proteins in here, and it will only take the assays from that. It won't take the whole library. So I specified that file. I can show you that. Is it done? Yes, it's done. In case the M/Z filtering, that might be an issue if you get the raw file from someone else and you don't exactly know what the settings were. At least for WIF files or like SciEx acquired data, you can just open one. And then if you press Control-I or Show Sample Information, then you see, you scroll here down, and down here you see the window definitions. So you see the windows are from 400 to 425. So 25 Dalton windows were used. And they were used like throughout the whole file. And it started at 400, and if you go at the end, it stopped at 1200. So you are only interested in peptides that fall in between that range. So 400 to 1200 is fine. In case you want to look at the MS2 M/Z range, you could scroll further down. I'll show you two ways. You can click here on experiment 2, which is the first SWATH file, and then it will tell you, does it? It tells you down here, start mass, end mass. So that one was acquired from 50 to 2000. So everything between 50 and 2000 is observed. But I told you on Saturday that the very low M-overset fragment ions are not so informative. So I would keep, in this case, I will keep this 350, and I would increase the maximum fragment ion M-overset to 2000. Because these high M-overset ions are quite informative, since they cover a lot of the peptide sequence. Down here, you have the number of fragment ions. Today we're going to use 5, since that particular library only contains 5 ions. Down here, you can do the same thing as I showed you with PeakView at the very beginning. You could say that you have actually a labeled data set, and you would like to have labeled assays. We have the usual SciLac labels in there. Theoretically, you can fairly easy add N15 as well. We did on purpose not add the tags, since they modify the peptide sequence, so the whole retention time is different, and as well as the fragmentation. So we just don't think it's a good idea to do that at this stage. That we first have to see like how different are they, and so not that people say, hey, I've done it, and then the library is crap and whatever. If you have in the final setting, you can specify the SWATH files that you have. This is in order to filter for the precursor selection window, that ions that fall in there are kicked out. You can either choose a file if you specify it yourself, and that's in the same way that you would use to like tell it to the triple TOF itself. So that makes it easy, because you only have to upload that particular library. It's also the same way it's listed here. So that's essentially a table like that, except here that column will be, you have to delete that column, but it's just start, separator, and then end of each of the windows, and that's it. But since we just used the 25 Dalton window and only, since we used the traditional one, we can leave it like that. And then you click on Generate Custom Library, and it's done. And you see here our 34 proteins. It used these for filtering. It has about 1,500 ions. These are all the transitions. If you divide that number by 5, you should have the number of assays that you have from many in the library. And then you can, on one hand, download the SWATH window file. So in that case, it would look like from 400 to 425, from 25 to 50, and so forth and so on. Or you can download the library. You click on that thing here. It does the trick, and now it downloads the library. And it's done. You can click on it, and then it should look like. And so you can here have a look at, this is the library as it's supposed to look like. Make it a bit smaller like this. So there you go. This is yeah, useful. This is particularly useful for the human library since it has over 140,000 assays. So it can make your life a bit easier. I will show you an issue, issues that you will run into if you only use that few of assays. I mean, these are just like 300 assays. This is not that many. I'll do it this way again because like this I can have a cheat sheet that I know what I should show you. Okay, so now I loaded this file where you see now there are 9 files and just a bit. Let me see if I have that in the presentation. Okay, so just a word to the data set that we look at. I don't know how many of you know about TB, but just a word or two. So this is a small, small. This is a bacterium, causes tuberculosis. There are surprisingly still about 9 million cases every year. That is, I mean, these are data from 2011, but it's kind of constant for some reason. The death, they keep on going down. India has quite a few, but this is just because you have a lot of people here. About 1 in 3 people is latently infected. That means they have the bacterium, but they don't have the disease. And this is a lot, especially because it can then, like, you can have essentially tuberculosis any time then, and then you're like spreading it as well. Very important are the multiple and the total drug resistant strains. And I mean, they started again to make drugs against it, but well, I've heard of like that there are already drug resistant strains against that. So this is it's really important to like do some work on that. That's why I started working on it as well. The genome is about 4,000 proteins, and you saw that we have that fairly well covered. And there are different disease models, and we're going to look at one particular case of that, but they're only like well established on the transcriptome level. What we specifically look at is a growth curve, and we have 3 time points: early exponential phase, late exponential phase, and stationary phase. And each of these we have in technical triplicates. They were grown under lab conditions, so so whatever some people think is like happy, I never know how happy a bacterium actually can be. Yes. The reason why I use that setting is because of that one. This is DOS-R is a transcription factor that's quite well understood. Stands for dormancy survival regulator. So it plays a role in dormancy. So whenever the bacterium comes in and goes in the macrophage, that transcription factor is turned on and puts the mycobacterium into hibernation. It expresses about 49 genes. The reason why we only look at, the reason why I selected 34 is because these are the ones that were identified in these runs, which is about 60%, which is like quite good. It is also activated in stationary phase, so we should see it in our data set, but we should only see it in the stationary phase data, like files, but not in the other 2. Or at least we should see an increase once it goes in once the MTB goes into stationary phase. All this information we know from transcript level data and not so much protein information. They've done some Western blot analyses, but these were done with like, I think I found information about 4 of the proteins. So it will also be tedious to do that for 49 proteins. Yeah, that's some results. Good. So you've done your analysis. The peak view finished, and then it basically just shows you like that. It doesn't do anything. what you can do now is you should be, first of all, I would save the session. Quantification, SWATH processing, save the session. And then what I would do is export the different data files. You can either do that for, or what you can export is the area, which is which gives you 3 files: the area of the ions here. So in that case, for each of these, the area, the summed area of these ions, which is the area essentially for the peptide itself, and then the area of the protein, which is just the sum for every peptide. Unfortunately, PeakView does that like pretty much the easiest way it could, and just does it like that. It doesn't like look for, say, if say a peptide is identified in one run and not in the other run, it only exports it for one. And then for the protein, it sums all the peptides up, even this one protein that was only found once, not in the second time. So there is some well, yeah, you can discuss if you really want to include that or not. I prefer looking at peptides that are identified everywhere. The next thing, what you could do is go to View Settings, and now you see there are a few additional panes you can activate that are pretty much only useful once you've actually finished an analysis. I'm going to click for now on the heat map as well as fragment ion response. We're going to look at some good results. What we're going to look at is at the spike in peptides. The spike in peptides, of these we do know they're in there. I mean, I've added them beforehand. So this is just a first control to like see that everything worked the way it should have worked. There we go. So we look at the IRT protein. The reason why, I mean, the retention time peptide up here, the one that I used to do the calibration, contains the same peptides. However, for the extraction, it only extracts peptide or proteins that have here this check mark. And the retention time calibration protein has per default that unchecked, unmarked. That's why it doesn't get exported. Since it is a duplication of assays, since we get the information for that protein anyway, I usually don't do it. What you could do is, in case you're only interested in a subset of proteins, you could go through the list and click on each of them. But that's, yeah, kind of tedious to do. And it is the same thing as if you just like filtered the library on SWATHAtlas level, import it there, and do it like this. Let's look at a nice example for now. You can, as you've seen, you can type in this search field and it will search the proteins, and you can type in a peptide down here and it will give you the peptide. And in this case, only like for the peptide sequence. You cannot filter for, that's not that. What you should look for in here. I just moved the pane over here that one gets a bit bigger. And I make that small. These windows are all like interactive. So you can go down here and like zoom in. You can also zoom in this range if you want. By double clicking next to the axis, you zoom out again. Like this. So this is how a peak group should look like. This is a good result. Since we spiked them in, it's not that surprising, but at least I mean, it's good to know that if you spike something in, you also see it there. This one here is also important. These are the areas of the individual fragment ions. And if you look at it that way, you see they should be fairly straight. That peptide should not change. You always spike in the same amount. And it couldn't change because of like different expression levels or different sample handling or something. You spiked in the peptides before the analysis, so they should be flat. You see here there's some variation, specifically here in that turquoise one, or whatever that color is. You see that's because here you see that shoulder peak. And so this is the file number 1. These ones here, for some reason they could just put numbers there. These are the files here. And if I go here to now number 2, which is this one, then you see that this shoulder peak was like squeezed over a bit. So there's some intensity that was added to the ion that we're looking at that actually doesn't belong there. That's an interference from another peptide. If you're not sure, what peak view allows you is click here and you see now there's shading, and it shows you what peak view considers a peak, where the peak starts and where it stops. Here you can choose the ion that you want, and in this case you see the entire peak was considered as 1. If we go back to the first one, so the one, if you look here, that's a bit lower, and now select this one, you see in this case the shading is different. So in that case, peak view considers this a peak and this one here a peak. So that's why you see that little difference. It's also normal to see here like some fluctuation. You will hardly see them like perfectly straight for all of them. Down here, this is a heat map of essentially that peak. So essentially, it's, instead of having traces, and you look at traces, what this gives you the information like on like every like a top view. Like assuming you look from here up here on that, and you only record the intensity as like a color code. You can have that either as absolute values, or you can have that as %. If you click the % button, essentially every ion itself has the maximum is 100%. Ideally, it looks like this, where you only have like one line there. You can modify the color here a bit and say like we want 90%, and now you see here this is really the only thing there. So this is the way that it should look, or yeah. But again, it's a nice looking peak. You can do the same thing for the % one. You can do that for this here as well. However, that one kind of always looks very noisy to me. Even if it looks kind of good in like absolute terms, this one here is kind of like, yeah. So I usually look at that one in this way. But yeah. Down here, what you see is again the spectrum at the apex. Below is the like expected intensities, which is the same thing as essentially your assay, like the relative intensities there. You can get them actually if you click here on that button, which is ions. There's a window that pops up and it shows you, okay, these are the ions and the intensities and M over sets. If you have more than 5, so if you have 20, you can actually add and remove some to see how they perform. But usually I don't do that because this is like, it's very tedious if you want to do it for a significant number of proteins. Yes, so this is one that looks a good example. One thing you can also check is let's add that ping as well. Let's remove the heat map and the peptide response. And so what you see here is, so this is the fragment ion areas, intensities listed. This one here is for the peptide. And now I filtered for one peptide sequence, which has 2 assays, so that's why you see here 2 lines. If I remove that, if I don't specify the peptide sequence, it shows all the peptides that have at least one significant identification. And so that one here gets much more crowded. What you can check as well is if you have one peptide, you would think that the dynamics across the different files is the same for the 2 charge states. What you will also say is that the retention time is the same, since you have the elution from the LC and then the charge gets added. So that's why the retention time should be the same. In this case, you see 74.06. And in this case, you already see this is a bit a lower and noisier region, but still a nice peak. And this is like 74.04. It's slightly different, but that's okay. The difference comes from that the higher charged ion is analyzed first. This is in a lower mass window, so this is first fragmented, and it takes a while, and then comes the higher charged ion. So that's why you actually see there a difference. Good. The other thing what we can test is how do bad IDs look like. One of the drawbacks here is you will get a you will get this data even if there is if the protein is not there for like not IDs. It's like if you would look at a shotgun spectrum with the probability of say 0.1. It won't look that great, but so in this case, in the library, there are it's the UPS standard. So that's a standard mixture of human proteins. They were not present, so we can just pick one. And now see, this is how that looks. So you see here in the fragment, most of them, this is just, it's not like nice and steady. It's not even like changing in the same way if it would be dynamic. This is just a mess. You see, in this case, you have the purple way up and this color again up here, and the blue is the lowest, and then here the blue is the highest, and the pink one is or the purple one is the lowest. So that's not good. I actually don't know. Since they were not spiked in that case for this protein, there was only one that actually managed to get below 1% FDR, this one here. So only this one is represented in the peptide pane, so that's not that informative anymore. What you can do as well is move around and see what the retention time looks like. This is 83.36 right now. And so you see here it's like 86. It just jumps from one location to the other. And you see already up here, there is no really nice alignment of the peaks. There's not really a maximum. You see here, these two are like offset already. And here only like the green is kind of in the middle and the blue and the pink is on the left. So this is not how a nice peak looks like. Again, if you know things like that, that's great. Usually you don't know what is not there. If you have these peptides in, assays for these proteins, it's good. And if you know that you haven't spiked them in, but it might get a bit harder. What I also like to show you is maybe not. Maybe not so nice example. This is RPOB. I don't know if how familiar you are with bacteria, but this is a RNA polymerase. So this is a protein that is just there. So if you don't find that, something is fishy. This is another good control. Usually, if you have like if you get the sample from someone, they usually know what kind of protein should be in there. But yeah, so here you already see the doubly charged one is again a nice high peak. This one, for instance, might be a candidate for these retention time normalization peptides. If you go to the triply charged peptide, you see that one doesn't look that nice. Down here, not too good. The peptide it's slightly off, but you see it's only off in like 3 of the samples. The other 6 are straight for the doubly charged and the triply charged. What you can see here is that one here. You can click here down, then you see this fragment ion Y3 has like spikes in all the stationary files, so in the files numbers 3, 6, and 9. And what you can do again is here, click down and highlight the area, the peaks, and you see now there is like 1 tall here, then 3 smaller ones, and there's another tall that is coming here. And if you look now in the file number 3, so this was the one that I just showed you, this is the one that is displayed now. If you go now to that one, what you see is here are the 2 tall peaks, and this one here is these 3 all together, and that's likely what has happened for all these 3. So that's why it might be better to exclude bad intensity, and then you see, okay, that would then like decrease these ones, and the area for the samples 3, 6, and 9, and you get a similar profile like up here. Also, I mean, the change here, this is maybe 15% or something like that. So depending how well the assays are and the data is, that might just be explained with the data itself. So it's not that huge change, but still. I think something similar happened here with the red one. You see here you have you have 2 peaks, and if you go to number 4, no, it might just, yeah. Maybe not that easy to see, but that one is definitely a bit higher than it should be. But yeah, so in this case, you also see, we can also check that one again. 75.28 is the elution, the retention time. Oh, I see it here. The retention time is perfect in these 2 cases, so they're fine. Like this, what I did with the charge states, you can do that in order to like gain some confidence if one charge state is very well identified and the other charge state is not that well identified. Other softwares like OpenSWATH and SWATHprophet give you the IDs per window that you acquired. So if you have an assay that falls in the overlap, you can you get the ID twice, and there you should see the same, something similar, so that one should be identified at the same retention time with the same areas, or like similar areas. But yeah. If you have the one with the two in the window overlap, you should also get rid of the interferences, because it's very, very unlikely that you have two peptides eluting that are that close together. It might still happen. One example that I like to show you is this one here. That's a bit of a problematic one. As you can see, I mean, it's from the same protein. We know the protein is in there. It's still from RPOB. It kind of has a nice peak for, or you see a nice intensities for two of the transitions, but the other three are very low. When you see in number 3 and 8, it's actually not identified, or it got the wrong peak. And what it happened in that case is, if you look at the in the ions file, when you sort by intensity, you actually see the top two. This is like 1E4 and about 5E3, which agrees well with this one here. That one is about twice as high as that one. But the other two are very small, and they're supposed to be very small. So they're like 3.5 e to the 2. So that should be 10 times lower than this one. And so there you might run into trouble in like that you don't identify 3 of the transitions, you only have 2. And now suddenly it's identified, in that case, I don't forgot. This is file number 4, so this one here, you see there is the protein is there, and in that case, it actually thought it's down here, simply because it probably took that one here because the other ions had at least some intensity there, and not like barely anything. But yeah, these are just like some where you might run into problems. I also understand that, I mean, in this particular case, you might say, I don't care, because that protein has enough peptides. We've seen some that perform well enough, and then it's okay. But if your favorite protein, or if, for instance, if you're interested in, okay, that doesn't have a serine, that makes it hard to argue. But if that particular peptide region is like modified, and you absolutely want to have that that peptide analyzed, then it's getting tougher. We've already seen an example for an interference. Let's have another one. We get to learn a new protein, RV0079. I have no idea what that is. So in this case, you kind of see, because we know it's in triplicates, I analyzed it in triplicates so that the samples here, number 1, 4, and 7, are the same. These are technical replicates, so they should show the same. So in that case, it kind of looks okay from that perspective here, but you already see that here the intensity of that from that peak might give you trouble. You also see that if you look at the fragment ion pane or area information across sample, it goes just down to 0, pretty much. What happened there? Oh, I was at number 3, I think. What happened there is think in this case it actually split. No. Oh, because I didn't change. No, I did change. So in this case, it still added some intensity to the peak group. In the other ones, it excluded it because it considered it as an interference, and that's something PQ does as well. If there is an interference, it will remove the ion and not export the area. One thing you can do for looking at interferences is you can look at the dynamic change, if it has a dynamic change. This one here is a peptide from the DOS-R regulator. And now you see in the two first files it's low and then it's high, and low again and high. So if you look at these ones, it has some like shoulder peaks or peaks that are close by that might give rise to an interference. If I go to the stationary file and look at that, you see that peak here increases, and that little shoulder peak kind of like stays in the same intensity range, so it's much smaller. So like this, you can also see like when a peak is actually an interference. Relative intensity should remain the same, which they do here. It's actually, I think, one of the nicer cases. So if you show here that the percentage, you see that looks much nicer, even on the spiking. Okay, one example that I want to show you, or one more example that I want to show you is the reason why you should always include enough assays. So if you go, no. If I type in pick, that's the protein encoded right before the transcription factor. This is, for some reason, it keeps the retention time protein up there, and even if you filter it, it just selects this one first. And now click on a different peptide because here nothing showed up. I thought maybe I have to activate it again. So now you see I've selected an entire protein, and it displays here all the peptides of that protein. What you can see here is that most of these peptides show the same profile as we've seen for the transcription factor. So for each of them, it's like low and then goes up, low and then goes up. Also, in that case, it looks that bad because that was actually not identified here. Let me pick a nicer example. There we go. This is the way it looks, and you see here like most of the peptides agree with each other. Something's wrong with that blue one. But for now, we don't address that. I mean, there you would have to look into that case and see why it's not that great. But in general, these peptides look good. What I do now is I close that session and I open a session where I only use these 34 proteins that we had before. And this is the same protein. The only thing we need is to add the peptide and the fragmentation pin. And it makes this. And now what you see here is suddenly that looks much more like noisy. There are many more differences. Like, it doesn't look as nice and clean. They don't agree that well with each other. What happened here is that in addition to the peptides you identified before, it had some other peptides that it also thought it would be good or are significantly identified. The reason for this is that this number here, that FDR value, is actually not properly calculated. I only had about 300 assays in here, which for a 3-hour gradient means in terms of decoys, you have about 2 every minute. That's not that much. You should have about 100 for 10 minutes, so about 5 times more at least. If you look here, let's pick one. I'm trying to find a peptide that actually does not look good. Here. So here you have a peptide. It has, so you see the observed detention time is like 20 minutes before the actual end, so that's probably during the wash or just after the wash. It has an FDR of 0, but it looks like this. So that's not okay. The reason why that happened is that PeakView specifically, I'm not exactly sure about the other softwares, but what it does, it only looks at the decoys around that identified assay. So in that case, it would take that one and take, again, like a retention time window around it, and only look at the decoys that are identified there. At the very beginning and at the very end, there are just no peptides there. And so it's very easy for a peptide like that to be called identified, but actually is not identified at all. So that's why it's worth having enough assays in there. And if you're not sure, look at it, and you will develop a gut feeling if this is really true or not. Also, you can test these things. So like, yeah, the retention time should be the same. You should always have around the same shift. The fragment ion intensities should remain the same, and so forth and so on. And otherwise, if there are like issues, but you think, hey, that's I think it's a true identification, but that fragment ion looks a bit fishy, you can also go in and like have a look. Did PeakView get that wrong, or